Hello everyone, and welcome to the IFST webinar, Meat Alternatives, the Manufacturing and Science Behind Mycoprotein, organized by IFST Easter Branch. My name is Anabel Muret Cabero, and I'm a member of IFST Easter Branch, and I'm a research scientist at the Quadram Institute. Today's webinar is about an alternative source of protein for meat replacement called mycoprotein. We will find out how it is produced and incorporated into products from Quorn. Moreover, we will understand the health and nutritional implications of mycoprotein through research studies. The topic of meat alternatives is clearly getting a lot of attention from the general public, and having this combined perspective of industry and research will help us to obtain a bigger picture. Next slide, please. So this webinar is being recorded and will be uploaded to the IFST Webinar Hub. Feel free to make comments and ask questions throughout the webinar using the Q&A function, which should be at the bottom of your screen. This can also be used in case of any technical difficulties. There will be a Q&A session after the presentation where we will respond to as many of these questions as possible. In this webinar, we have two speakers. First, I would like to apologize for the changes in speakers representing Quorn Foods. Tim Finnegan and Holly Roper couldn't make it today, but we had the pleasure to have Tess Kelly. Our second speaker is Raffaele Colosimo from Quadram Institute. So now I would like to introduce these two great speakers we have today. Tess Kelly completed her master's degree in health psychology from the University of Nottingham, focused on plant-based nutrition. Tess works at Quorn Foods as Sustainable Development Manager, leading specialist communication on health and environmental projects. Our second speaker, Raffaele Colosimo, graduated with a master's degree in Human Nutritional Science from the University of Pisa in Italy. He is a final year PhD student at the Quadram Institute, working on the relationship between mycoprotein structure and human digestion. His work has been really productive, resulting in 5 peer-reviewed articles already published. For more information on their biographies, please have a look to the IFST website. So with that, I will hand over to our first speaker, Tess Kelly.
Thanks, Annabelle. It's an absolute pleasure to be with you today. I'll just share my slides. So, as I say, it's a pleasure to be with you today, talking about certainly my favorite topic, and that is the magic of mycoprotein. As Annabelle rightly said, the rise of popularity and interest in the science behind meat alternatives is more popular than ever. And we really represent a unique and very unique composition in terms of protein sources. So I'd like to give you a bit of an insight into that unique offer of mycoprotein, how it's made, and our philosophy and evidence base behind the health and nutritional benefits. So why is mycoprotein unique? And we actually start from a very different place for our protein sources than animal-based protein, of course, but also the majority of other alternatives on the market. So a lot of people are surprised to find out that actually mycoprotein is derived from a fungi, and fungi are biologically distinct as a kingdom from both animal-based proteins and plant-based proteins, and actually they are more biologically similar to animal proteins. So a lot of people associate fungi with mushrooms, and actually the mushrooms are sort of similar to the fruit. They're a fruiting body of fungi, and you'll see at the bottom left-hand side there the main proportion of fungi in our kingdom is actually a mycelium. So that white strands you can see in the soil is something called mycelium. And that is the main body of fungi, and the majority of fungi look like this. And this is actually what our microfungus looks like. And that is what we develop our meat-like texture from. So fungi is a third kingdom. Why is that even important? We know that we are facing some very, very significant challenges in terms of our population growth, how we're going to feed everyone a nutritious and a sustainable diet. And actually fungi is a new food source to introduce that diversity and that versatility into our diets is a really exciting opportunity. And we have been producing this food since back in the 1960s is where it all started. And we have been providing Quorn products onto particularly the UK market since 1984. And now we sell products into over 20 markets worldwide. It's an extremely efficient method of producing a whole food source. We actually use the technique of fermentation, which is an age-old technology in earnest, but we apply a kind of 21st century scalability to that technology. And I will take you through a little bit about how that's achieved and some of the key health benefits and evidence that we continue to invest very, very heavily and take very seriously. So one of the most interesting elements that we are investing in more and more in recent years is understanding the fiber within our fungi and our mycoprotein. So as we all know, fiber is one of the key nutrients that most of us are not actually eating enough of. And when we talk about meat alternatives, or when we see the general conversation around replacing some of our animal protein consumption with alternative proteins, is actually the protein side of things, and that is very important. Our protein source is complete and contains all 9 essential amino acids, but also we have a unique source of fiber that I know Raffaele will be talking to you about a little bit more in detail. But we know that from the animal protein side, animal sources tend to contain 0 fiber or very little fiber. Plant-based sources are a fantastic source of fiber when they are in their whole food composition. But interestingly, what we tend to see in the meat alternative category in particular is the presence of isolated proteins. So this is where the protein is kind of extracted and refined and texturized to become a meat analog, if you like. With mycoprotein, we don't have to extract that protein. We actually use the whole food source. So from that fermentation process, we take the whole food and use that within our products. And so we retain that amazing fiber content. And the fiber within our products is 2/3 soluble and the rest is insoluble, and it has a particularly interesting fiber called chitin as well, which is again unique from plant-based sources. And actually, in fact, our over 90% of our range has a claimable fiber content on pack for either a source of or high in fiber, and we're really proud of that. And it continues to be a key metric for us developing new products. So how is Quorn actually made? If you look at the top picture with that little piece of card in somebody's hands there, you might be able to just make out a tiny, tiny speck. And that's actually the spores of the fungi, which are the same as seeds in the plant world. And we start there. That's like a peppercorn-sized lump of spores that we then take and we add that to a sugar solution. So we used glucose, and the fungi feeds on that glucose, and we pamper the fungus. We give it all of the right conditions to grow and to develop at scale in terms of its sort of nutrition and composition. And underneath you can see, I believe this is actually one of Raffaele's pictures, which is just astonishing to see this at a microscopic level. And what you see there in the pink is actually the sort of core protein, and the blue outside is its fibrous shell. So this is where you can see this whole food structure. And then we bind the protein in our vegetarian products. That is is achieved by using all free-range egg white. In our vegan products, we use a potato protein as well. Once that's bound, we use steam cooking to start to bind that product together. Once it's chilled, we then use a very specific freezing technique, which allows ice crystals to grow at a controlled rate and further push those fibers that you saw in that first picture together and create that meat-like texture that you see when you break into a nugget or a fillet, that really mimics the texture and the taste of meat alternative products. So some of the key highlights for Mycoprotein that we're really proud of is that, as I said, it's a complete source of protein containing all 9 essential amino acids. It contains no cholesterol, and we have some really fascinating research looking at the ability of Quorn Mycoprotein to reduce blood cholesterol. It's high in fiber naturally. As I've said, it's low in total fat and saturated fat, and because it's a whole food, as I said, we retain some of those really key minerals and vitamins that are crucial to our overall metabolic health. And most importantly, where we end up is a core protein source that enables us to make really delicious and versatile products that cover red meat all the way through to some exciting kind of fish innovations and particularly good chicken products if you've tried those. So just again, a very quick highlight on our sort of key health benefits that we have researched over a number of years. We have a 35-year research history. It's a complete source of protein, but more importantly, that protein is very bioavailable to the body. You may have seen in the last couple of years through our research with Exeter, we've been looking at the muscle building capability of our protein compared to gold standard animal sources such as whey protein. And we've actually discovered that by consuming Mycoprotein, you can actually build skeletal muscle twice as fast as milk protein and some animal sources as well. So this is really exciting, and it's also kind of groundbreaking in the world of understanding the potential for alternative proteins to contribute to longevity and particularly healthy aging. We've talked about fiber, and Raffaele will go into this in a little bit more detail, but we're particularly interested in the gut health benefits here and insulin resistance and the contribution of that fiber to some of these more complex metabolic challenges that we face. It's low in total unsaturated fat and contains no cholesterol. And again, some of the research at the University of Exeter looking at the potential for reducing, particularly LDL and your bad glucose, as it gets termed, is particularly exciting as part of a balanced diet. And as I said, it's a source of key vitamins and minerals, in particular choline, which is again usually found in highest levels in animal protein. This is why we think Mycoprotein is truly a magic super protein as a choice. So as I mentioned, we work with a number of leading universities and research institutes, and this is something we're incredibly proud of because we do have a unique food source that we've discovered. We also take the responsibility of that extremely seriously and always have done as a business to really invest and understand the key benefits of our foods on human health and planetary health. So with all of our research, we operate on a no strings attached basis, which means that no matter what our researchers find, they publish it, and that's extremely important to us and to the universities and institutes that we work with. But we're finding some really exciting results. This is a snapshot of some of Alistair's data you can see there at the top from Exeter, looking at rested and exercised muscle building. You can find much more detail about these studies on our website, Quorn Nutrition. And we really sort of broke the mold here in terms of actual knowledge and insights and understanding the role for non-meat alternatives to play a role in both athletics and sort of building muscle in younger people, but we're also looking now at adults of an older age and in the community as well. So extending this research is very, very important and a priority for us. But as I said, we go so much further than just protein. Increasingly, we are looking at fibre and gut health with a range of universities, and you can see Raffaele down there, who's going to be talking through some of his research. But we also look at cardiometabolic health in totality, so not only looking at your protein, muscle building, fibre, gut health, but also your cardiometabolic effects. So whether that's looking at insulin resistance or, as I said, cholesterol levels. So we have a huge amount of research already published on this, which I think we have a snapshot of just here. As I said, it's been over 35 years of research, and you can see in particular in the last couple of years, the number of publications in peer-reviewed journals that are coming out of our research community and outside of our own research community as well. So the World Economic Forum recently published a health economic analysis of the effects of replacing a portion of animal protein for mycoprotein and peas and legumes and soy, and mycoprotein came out as one of the kind of top protein sources to reduce incidence of disease and risk of death. So all of this history is really starting to come to the fore in terms of the wider landscape of research on meat alternatives and alternative proteins, which is really exciting to see for us, certainly. And we continue to invest in this, and it's certainly one of the parts of my job that I find extremely rewarding, and getting to work with such talented scientists as well. You can find more on this. We've just published this timeline in its totality on our website and on our Instagram as well, if you want to dive into some of those references. What my team really do then, once we have all of this information and data, it's then our responsibility and our great pleasure to take all of that and turn that into exciting and engaging and helpful communications, in particular for healthcare professionals. So we work very closely with a range of nutritionists and dietitians, both in industry and in clinic. We partner with both the British Nutrition Foundation, but also we're a strategic partner of the British Dietetic Association. And that enables us to also have that kind of external lens of our research and really understand the challenges that healthcare professionals have in terms of guiding their clients through questions about the role of these products in their diet and lifestyle. But we're also able to bring to life, the science into really exciting food and cookery and food literacy projects in the community and virtually. So one of our flagship projects last year was something called Sustainable Eats. We know that dietitians are also, not only really keen on understanding the health benefits of our products, but also the environmental benefits. And this project was really successful. It's been received really well, and you'll see a lot more about this later this year. But it was a recipe project looking at how to incorporate mycoprotein into really familiar recipes that we see in many homes across the country, and what the actual carbon and nutritional impact of making those incremental changes can really achieve. We also do a lot of work in communities. You'll see on the bottom left-hand side there, that's one of our home economists, and we go into many schools across the country to teach basic food skills, whether it's learning how to chop vegetables, baking bread. A lot of the time, we don't even focus on Quorn, and we believe that is a core foundation of building healthy lifestyle habits, and getting kids interested and confident around the food that they eat as well. So this work all contributes to our vision as a business to become a net positive company beyond the impact of our products, but also in terms of what we do, the partnerships we build, and the communities that we serve as well. And it's just fantastic to see kids' reactions to some of these classes. Where can you find out more about all of this research and the projects that we do? I work very closely with a team of nutritionists, and together we have built a resource dedicated to healthcare professionals or interested nutrition scientists and budding healthcare professionals. So we have a website where you can dive into lots of resources and fact sheets. We're building more courses in the future. We do a lot of kind of webinars, and we have an Instagram channel as well, where you can dive into a little bit more detail in terms of the key benefits of our products, but also any events that are coming up, including we also have a newsletter as well. So if you want to keep up to date with any of this, please do sign up. And I think that's everything from me. So thank you for listening. I really look forward to taking questions. And now I will hand over to an expert in mycoprotein beyond myself, and that's Raffaele. So thank you very much.
Thanks, Tess. And thanks, Annabelle, for the kind introduction. Thank Tess for this also introduction on mycoprotein. Very nice. So I can now go straight to the science of my PhD project. And the title is Understanding the Digestion and Health Effects of Mycoprotein-Based Foods. So as we been mentioned before, that several in vivo studies have shown that protein is highly bioavailable, and also a consumption of mycoprotein has been associated with reduced insulin and glucose postprandial responses, and also to reduce the blood lipids. So in order to understand what caused that, we have to know what we are working on. So mycoprotein has been mentioned that is made by fungi, produced by a process of fermentation. And we have this fungal hyphae, which the single unit is a filamentous cells. In terms of macronutrients, we know that mycoprotein is high in protein on a dry weight basis and low in fat. While fiber, the dietary fiber, is an important component, which has been associated with health effects from the literature, and it's also important because it determines the structure of the cell wall. The fungal cell wall, in this case referring to mycoprotein, differs from the cell wall from plant-based foods, in which the cell wall of fungi is mainly composed by beta-glucans and a single layer of chitin close to the plasma membrane. Then there are also some glycated protein intercalated in the cell wall. While the plant cell wall is mainly composed by cellulose and also pectin and beta-glucans is shared between the two kingdoms of life, between fungi and plants, but it changed the link. So the link from fungi is beta-1316, while for plant is 1416. And this can also affect the digestion and the health effects promoted by this type of fiber. Moving through the methodology of my experiments, so the main sample used for my experiment was mycoprotein in form of a freeze-dried powder. So it was a pure mycoprotein, it wasn't the final Quorn product. And most experiments involved the simulated digestion of the human intestinal tract following the InfoGest protocol. In this protocol, the digestion is divided in different steps, so the oral phase, gastric phase, and the intestinal phase, in which we control the pH, temperature, and the mixing and the addition of enzymes to digest the nutrients. Several time points are recovered from these steps, and then the analysis involved several assays for the analysis and quantification of protein, carbohydrates, and lipids. Then there is a microscopy to visualize the hyphae, the cells, in confocal, bright field fluorescence. And laser light scattering is showing this presentation for determination of the particle size. Other techniques that we use regularly in the lab involve the characterization of rheological properties such as viscosity or determination of protein hydrolysis with electrophoresis. So moving through the first area of investigation, which is the high protein bioavailability. The experiment I carried out involved the use of mycoprotein in two different concentrations, one at high concentration at 25% in water or low at 10%. So first of all, what I measured was the particle size of mycoprotein just incubated with water. And then treated with different physicochemical methods, in which involving homogenization, glass beads grinding, or sonication. What we observe is that the sonication lead to a reduction of the size. But what we want to understand if this also can impact the protein release. So in the next experiments, the incubation just with water, which is the control, which are the bar in white and gray, we measure the protein release, and it wasn't different between the several treatments, the glass bead grinding, homogenization, and or sonication. So the next step was to add different buffers such as Triton, which didn't see a high increase of protein release, while the highest release was observed with the urea buffer plus incubation and sonication. And the protein was observed around 30% in vitro. So we wanted to understand if this can be compared to the simulated digestion, the in vitro infogest digestion. And what we obtained here was a digestion of gastric phase and intestinal phase, in which first of all, we noticed that in the first 2 hours, the digestion of protein wasn't that high compared to the intestinal. And again, the mycoprotein at 10% or the 25% sonicated or not, so reducing the size before the digestion, we observed that actually there was the size, the reduction, it wasn't the reduction of the particle size, but it was the concentration that lead to a highest release of protein in the intestinal step, and was even higher than the incubation plus sonication with urea. Then we wanted to observe with microscopy what happened through these different steps, and what we observed first with lactophenol cotton blue dye, it was that the gastric phase, the cells look full, which are completely stained, while in the intestinal step, the cells appear releasing their content. And similarly with the fluorescence microscopy, in which in pink the protein and in blue it's the cell wall, we observe a partial release of protein in the intestinal step that was in line with the in vitro assays. So the conclusion, we noticed that the enzyme digestion in this case was better than the physicochemical method, and also that the intestinal step was crucial for influencing the release of protein. The next area of research involved the reduced insulin and glucose response. So considering these experiments, we wanted to understand what was happening, what was causing this effect. And first of all, mycoprotein was digested in an intestinal step following the infogest method. And the digestion of the internal glycogen, which is the source of carbohydrate, was compared to free glycogen. It was an extracted glycogen without cell wall. So what we observed, it was a similar kinetic, but was lower. That may be due to the presence of cell wall that slowed down the digestion. And this was carried out through 2 hours of the infogest protocol with the alpha amylase at 200 units milliliter. Similarly, with 400, the double of the activity, the kinetic was in line with the 200 units milliliter, and we observed a slower kinetic for mycoprotein. And this led to a complete digestion of the glycogen. This experiment was also carried out because we wanted to remove the source of carbohydrates from mycoprotein and use the glycogen depleted mycoprotein for the next experiments, in which mycoprotein that was depleted in glycogen was incubated in a solution of starch at 5 milliliters. And this was digested with alpha amylase, which is the line in red. Then adding several concentration of mycoprotein, 2.5 from 20 milliliters, we observe a decrease of the starch hydrolysis. So this suggests that adding mycoprotein, this reduces the starch digestion. And also we observe that by adding increasing concentration of mycoprotein, the more mycoprotein you add and the more amylase is removed from solution and bind to mycoprotein. This led to investigate through confocal microscopy to the diffusion, this binding of the amylase with mycoprotein. And with the stain of fluorescein or chalcophore white, which stain in green the protein and the internal content, and in blue the chalcophore white the cell wall, we use a label enzyme which was the amylase that was labeled with fluorescein, and we saw that the enzyme is in some spots of the cells. And thanks to the Z-stack projection, we see in an orthogonal projection the amylase that was conjugated with fluorescein that was inside the cells. This suggests that the amylase can diffuse through the cell walls, and diffusing through the cell walls is removed from solution, and this can lead to a reduced starch digestion. And this consequently in vitro can slow down the digestion of carbohydrates and have an impact on glucose homeostasis by reducing also the insulin release. The next step area of research is the blood lipids reduction. So we know this from the literature, and we want to understand the mechanism similarly to the experiment with amylase. In this case, we incubate mycoprotein at 10, 20, or 30 milliliters in an oil and water emulsion, so a source of lipids that was digested in 1 hour of the intestinal step with pancreatin, which contained the lipase and colipase to digest fat. Also in this case, the concentration of mycoprotein reduced the lipolysis, so limit lipid digestion. But this is not the only effect that mycoprotein can have on lipid digestion, as also it was observed in the conjugation with bile salts, in which in an intestinal step with only pancreatin, adding mycoprotein didn't lead to any effect. But while mycoprotein was before incubated during the gastric step and digested, and then in the intestinal step, there was a binding of bile salts that were depleted from solution. So this was interesting to notice that a gastric step was promoting these effects. So we hypothesized that the gastric step, the enzyme or the pH could cause this bile salts binding. So the mechanisms of the bile salts were investigated in a digestion without enzymes in this case. And the intestinal step, again, didn't lead to any binding, while before incubated with the gastric step lead to this effect. So that's what we define the gastric step effect, that is potentially promoted by the gastric pH that is able to modify the structure or activate the cell wall to bind the bile salts. The conclusion from these studies is that mycoprotein can bind bile salts and also limit lipolysis, and this can influence the lipid digestion and lead to a blood lipids reduction after mycoprotein consumption. Just a general summary of these results. What we observe is that the protein bioaccessibility, that the enzyme that digests protein can diffuse through the cell wall, and the main protein hydrolysis, it happens in the intestinal step. Then the enzyme alpha amylase can be sequestered within the mycoprotein cell walls, and this can lead to lowest slow carbohydrate digestion that can influence glucose homeostasis. And last, the mycoprotein that can reduce the lipase activity and bind bile salts and influencing lipid digestion. These results are important for advancing medical knowledge and understand how mycoprotein is digested, to develop new strategies and also help to reduce the risk of food-related diseases such as cardiovascular diseases and type 2 diabetes. And with this I conclude. I would like to thank my supervisory team and all the people that helped me through these experiments and through this journey, and thanks all of you for listening.
Thank you very much both for your really interesting and excellent presentations. We are now moving on to today's Q&A session. So I invite the speakers to join and switch on the microphone and camera. So just a reminder that you can submit questions using the Q&A function, which is at the bottom of your screen. So we have received a lot of questions. So about the process itself, so one question is how do you know how hard it is to grow or obtain the spores? Tess, do you know?
Yeah, it's an interesting question actually, and fungi are very are interesting in terms of the kind of self-replicating, if you like. So if you imagine kind of having you know, a sourdough starter, if you like. There's almost like a mother culture that we keep, and it's related to the originally discovered fungi. So we don't have to go out and kind of, you know, keep digging up soil to find more of the fungus. It's a self-replicating organism. So that again is yeah, super interesting. It's under lock and key.
Yeah, that's great. So, and now we have several questions about the nutritional profile of Quorn products. So there is one question asking why there is some nutritional difference between Quorn in the UK and in other European countries.
Yeah, it's a really interesting question, actually. And I think it's something that I will, I know it's from an anonymous attendee, but I'll come back to you with a bit more detail. My understanding is that for some of our products, obviously there will be kind of taste preferences that we will want to take into consideration within various countries. So we have sort of sensory taste panels for each of our markets. And so there may be kind of preferences that we take into account. Also, we have to consider products traveling to those countries, so there may be shelf life considerations as well. So we'll have to tweak the recipe just slightly to be appropriate for that extended journey. But I can certainly speak to our product development team and come back to you if you've got particular products in mind as well.
That's nice. So one of the attendees was asking about chitin. So Raffaele, can you explain a little bit more about that and the difference, for instance, with the plant cell wall?
Yes.
I know that you already explained a bit in your presentation, but if you can expand a bit.
Yes, well, as I said before, it's this single layer that is close to the plasma membrane, and well, it's only in fungi, or well, in shellfish in this case, but not in the plants, in the plant cell walls in this case. So it's different from beta-glucans because it's not branched, so it's a single chain of that is close to the plasma membrane. And well, in terms of digestion, it's interesting because it's not so present in our diet. And there are some evidence actually of this human mammalian chitinase that is present in the stomach of just some individuals. But we don't know actually if this can influence its digestion. And it's an important area of research, actually. It's really interesting.
That's good. Okay, so we have another question about salt content. So how challenging is to reduce the level of salt in your product, and how have you approached this? I think that is an important question.
Yes, yes, it absolutely is, and we're very cognizant of the role of salt in sort of health and you know, prognosis of particularly cardiovascular disease, of course. We conduct nutrition profiling across our entire portfolio, so we tend to look at the overall profile of our macronutrient content and how that contributes to an overall score of healthiness, and that's our kind of main metric. But also when we look at the number of products where we actually do have, you know, a red salt value on pack, it's actually less than 10% in terms of all of our products. So within the product development process, it is you know, a requirement that we aim for a non-red on pack. Where those products do have a red on pack, we ensure that those products are not, you know, overly marketed. It tends to be if it's a you know, a specialist product rather than one of our main ranges. But it is a big challenge. We're looking at you know, low sodium alternatives in particular. One of the big challenges is on texture. So as soon as you introduce a slightly different ingredient into the mix, it can change that kind of texture that people are looking for. But you know, in terms of our responsibility there, it is very, very high on our agenda, and I know our development teams are looking at what alternatives that are out there, including you know, seaweeds and all of that good stuff. So again, I can go back to some of our technical colleagues and to answer that question in more detail if anyone would like to get in touch.
Yeah, that's great. And another important aspect for the vegetarian vegan products is vitamin B12. So what is the source of vitamin B12 in mycoprotein?
Yeah, another great question. So mycoprotein itself, in theory, doesn't contain B12 naturally. However, B12, rather than coming from animals, as I'm sure this audience knows, B12 comes from bacteria in soil, and that's where it's kind of originally manufactured. And so we do get a slight B12 source through our glucose supply. So it's not something that we can very easily regulate. So the B12 content does vary very slightly within our batches, depending on that glucose source. But it's something that we're investigating and seeing whether we can introduce a more kind of natural fortification and a biofortification process, rather than, we see a lot of companies obviously fortifying by adding B12 and iron in powders into the mix and having to go through another stage of processing. That's something that we are trying to avoid, and that's why you don't see a lot of our products actually actively fortified at the moment, because we're looking at solutions where we can encourage the fungi and the mycoprotein dough to take up sort of natural sources of B12 within the original fermentation. So there's some interesting projects going on in terms of, whether it's introducing a kind of an additional yeast or an additional source within that broth mix initially in the fermentation process that can give us that natural B12 hit. So quite excited to see where some of those trials go.
Yeah, that sounds really promising. We have another question for Raffaele. So your studies are focusing on bioaccessibility. So can you explain for the audience what is the difference between bioaccessibility and bioavailability?
Sure. So basically, my studies involve bioaccessibility because it's more related to when the protein, in this case, or nutrients, is released. So if we talk of protein bioaccessibility, it's in the stomach or in the intestine when the protein are released from their matrix, and it can be digested by the enzyme. Then bioavailability, it's more in vivo. You can observe that when you observe the absorption, the metabolization, for instance, of the nutrients. So the first step involves the bioaccessibility release, and then bioavailability when it's released and absorbed.
Yeah, that's great. So moving on to other question. So it's about unprocessed mycoprotein. So have you tasted unprocessed mycoprotein? Have you?
You know what? When I saw this question, I was hoping it was going to be asked, because actually I have. And we actually were visiting some of our research team in Exeter, and one of the studies there is actually looking at sort of controlling cooking methods across lots of different protein sources. And so we're taking that core mycoprotein in its dough-like texture, adding some herbs and using that. So I did actually get the chance to taste it. Obviously it had a herby aroma to it, but it wasn't bad actually, because it's fungi based. It does have a natural kind of umaminess. Obviously a bit of a kind of a mushroomy taste, but the texture itself is more kind of paste-like. I would liken it to a pate. But yeah, it wasn't bad, I was pleasantly surprised.
Was it in a form of a sort of burger or something like this?
Yeah, I think it's going to be served in sort of a patty, but I just got it. Yeah, just scooped it, put a spoon.
Well, definitely better than just a powder, I guess.
New experience for me, certainly, but no, I wasn't mad at it.
That's good. No, because I saw it in the lab working with Raffaele, and it's kind of powder form, but I guess that if you put it in a different processing, you can have different textures.
I also got some kind of pure mycoprotein as in form of burger plates.
Yeah.
So that looks definitely better than just the powder. But I haven't tried, I have to say.
So talking about new products or development of new products of Quorn, have you tried to do snacks, or have you considered to do snacks of mycoprotein? Yes.
Yeah, I mean, we do have a snacking range. I suppose, depends what you mean by snacks. So, you know, we have, you know, the former kind of picnic eggs. We do sort of small cocktail sausages as a more kind of grab and go option. But certainly, I think, as you said, you know, we can kind of provide mycoprotein in a range of textures. So that powdered form that we use for some of our studies, you know, might be applicable within, you know, a high protein cracker format, for example. And actually, because, you know, we've been around for a long time, I know there've been loads of experiments in terms of, you know, applying mycoprotein to even ice creams and, you know, noodles and all kinds of formats. So I would say kind of watch this space for kind of what new formats you might see mycoprotein appearing in, but at the moment we focus on the kind of the alternatives to your regular kind of meat snacking options. Yep. But there are some out there, and I'd highly recommend giving particularly the cocktail sausages a go. They're delicious.
And what about liquid in drinks, for instance? Have you considered to put mycoprotein in drinks, beverages?
Yes, again,
really
I think we've experimented with kind of myco milks in the past as well for some of our studies, particularly on kind of protein anabolism and muscle building, to kind of give the same sensory experience as taking a whey protein shake. We have turned mycoprotein again into a kind of protein shake format that's been, you know, chocolate or vanilla flavored, and actually the feedback from the participants is that, you know, on the whole it's actually quite palatable, particularly considering in the sort of vegan protein shake world, you tend to have a lot of pea protein, and some of it can be quite grainy or have quite a harsh aftertaste. So it's certainly very different, and it's you know, an interesting pipeline for us, but you know, obviously you know, a big investment and one for further down the line.
Yeah, we have another question about if it's too much, or can you have too much protein in your diet, and what is the daily tolerance amount? Do you know if we have a limit?
Yeah, again, it's a really interesting one. It's a hard one to define, really. I don't know whether, Raffaele, you know of any kind of protein tolerance tests. I mean, ultimately, we don't want to be consuming too much protein in general. Mycoprotein is a little lower in its protein content than animal sources anyway, which means on a protein consumption level, you could consume more. We tend to only put limitation recommendations into food service, in particular schools and colleges, particularly based around kind of the fiber content. So particularly if children aren't used to consuming a lot more fiber, it can cause, you know, it can be you know, a bit much, and we don't want to also restrict people's diets. So diversity is the most important you know, indicator really in terms of a healthy diet. So we tend to recommend on school menus in particular that children aren't having mycoprotein or Quorn-based products more than twice a week, so that it creates the room and the space for other you know, vegetarian options or animal-based options as well. So there isn't a kind of scientific limit as far as I know, but again, I'll double check on that one for you.
That's great. We have, I think, time for one question more. So, Raffaele, following up your answer about chitin, there is another question asking, do you know if humans metabolize chitin well, or does chitin benefit our microbiota more? Do you know anything in that aspect?
As I was saying before, it's like there are some evidence of the human mammalian chitinase that some people can already digest that, but this can vary in the population. In terms of fermentation, actually, this is also, I cannot reply for sure, because actually it's something that we are currently investigating to understand the digestion in the upper GI tract, and then the fermentation in the large intestine, in the colon, so how the microbiota can ferment the source of fiber of beta-glucans or chitin. So this is still an ongoing process.
Good. So I think, yeah, I think we have time for more questions, actually. Let me see. Oh, yeah. So mycoprotein is a sustainable product, but one attendee is asking what happens to the spent fermentation broth industrially. Do you know about that, Tess?
Yeah, good question. So yeah, obviously we use a lot of water in our process, and that is kind of centrifuged off. Again, there's some really interesting kind of substrates that are within that broth mixture. So we have been looking at taking kind of evaporating that, taking those sort of glutamates and some molecules that really have those umami sort of overnotes and flavors. So we have a kind of trial product that we've been testing for a number of years that's called Mycoscent at the moment, which could be used as a seasoning or could be sort of added back into our own products. We've done some trials that are super interesting in terms of being able to, again, on your salt question, reduce the amount of salt in ready meals, on even crisp seasonings by about 30%, because it replaces that need for the addition of sodium with a more umami, kind of satisfying, rich mouthfeel and taste. So there's an interesting project there. But in terms of the actual use of the water, we absolutely we clean the water completely, and it's returned in a pure format to the teas and to the water supply. So we remove all of that additional kind of mycoscent substrate at the moment for sort of development and testing at the moment, and the water returns to the supply as clean. But we are looking at kind of circulating that back into our fermentation without doing that cleaning process, and seeing if that again affects the fermentation outcome.
Yeah, so that follows up with the other question: is the fermentation process a waste-free process?
I would say it isn't completely as yet. I think it's got high potential to be a circular process, and again, that's one of the reasons why we're looking at that circulation. I think we can't, because we clean the water and we return it back to the teas, we can't claim that that's necessarily waste-free, because we do remove those substrates from it at the moment. But if we manage to kind of crack either this Mycoscent project or recycling that water, then it should become a waste-free process. Of course, it depends where you put the parameters of waste there. If we include the farming end and the wheat supply, the glucose, or further down the chain, there are slightly different issues to consider there, but the fermentation in and of itself is very, very low waste.
Yeah, that's great. Maybe another question for Raffaele: what is the PDCAS of mycoprotein? I guess that should be something similar to milk, right? The score
it was
of the
tested,
protein.
yes, in the past, and was actually one of the best. It was around 1, because also for mycoprotein per se, but also then the final product, adding the egg whites, it reached really a high level. It was one of the highest, even more than meat. But actually the PDCAS has been replaced recently, well, 2013, from the DIAAS. So that, which is another method, like more up to date for the determination of protein digestibility.
Yeah, that's great. Okay. Okay, so yes, the last question. Let me see, it's so difficult to choose one. Okay, so coming back to we already more or less answered that. So has anyone other than Quorn tried to launch a mycoprotein? If not, is it the bar? What is the bar to development entry?
Yeah, so again, really interesting question. So we don't have a patent anymore on mycoprotein, so other companies are free to kind of utilize the same fungi and the same process. We've seen a lot of experimentation coming from Israel, interestingly, and also from a company in Scotland, I believe, are about to kind of set up a business to business model of mycoprotein, which is exactly the same fungi as ours. So it's certainly a growing area in the alternative protein space. In the US, there's also a couple of startups which are looking at very different organisms still in the fungi kingdom, but will be sort of producing a type of mycoprotein as well. So it's the barriers to development are really in terms of the sort of scale and the technology. So the fermenters that we use are, 30-foot high. They're incredibly specialized at what they do, and it took us 20 years. This is back in the 60s, of course, but we spent 20 years of research and development and finessing that technology, and millions of pounds spent really speculatively, not knowing whether this is actually going to be feasible, which is a crazy thing to think about now and doing that now. But the level of investment in some of these startups is very, very promising, and I think you will see a lot more versions and types of mycoprotein coming onto the market within the next 5 years.
Yeah, okay. Well, thank you very much for this session. It was great for the talks. Thank you a lot. And thank you everyone from across the globe for attending this webinar today. If we didn't manage to answer your questions, please contact our speakers directly. Their details should be on the screen now. We will share a recording of the webinar, including the Q&A session, to everyone who attended today via email, and on our our online webinar hub at www.ifst.org/webinars. So before you log off, please fill in our short survey, which will launch after the webinar ends. So thank you very much again to Tess and Raffaele and everyone for attending. Thank you and goodbye.